Hello students, in the last lecture we have discussed with the amino acids and the proteins. I have started with the amino acids and proteins, and before discussing with amino acids and proteins, even I have discussed with the major energy fuels present in our body. And we have also seen their maintenance, their absorption and their storage. So in the last lecture, I have discussed with the carbohydrates, and I have discussed even with the proteins and amino acids, and also regarding the lipid molecules. So I have given a general introduction to how the body will be mainly keeping the reserves of the energy, and also the energy which is mainly used for different physiological purposes that are mainly derived from the carbohydrate molecules. So in the last lecture, we have discussed with the importance of the energy fuels and the primary energy source. We have discussed with the carbohydrates. Primary energy source, it is mainly the carbohydrate molecules, and mainly the storage reserves are present inside the body in the form of the lipids, which are stored as triglyceride molecules. And even I have told you that the proteins cannot be stored inside our body. They are slowly digested, but still the carbon, the nitrogen part of the proteins will be converted into urea and ultimately released in the urine, and remaining part of it will be converted into glucose. The two main points which we have discussed in the last lecture, it is being the usable energy, that is present inside the system. Anything that we will be taking from the food, it will be converted into glucose to produce the energy to our body. And anything that has to be stored inside our body, that will be converted into fat. So excessive amount of carbohydrate, excessive amount of the protein, whatever it is excessive for our body, that will be stored as the fat. Anything that we will be consuming, that is mainly converted into glucose in order to provide energy. Okay, so this is what we have discussed in brief, and next we have discussed with the amino acids and proteins. I have told you the proteins are the macromolecules, and they are the part of the acid insoluble fraction. And what are the building blocks of the proteins? I have told you they are mainly the amino acids. All these amino acids, these are repeating units, these are monomers. They may be different type of amino acid, but still all these amino acids are repeatedly, they will be attached and they will be forming the complete macromolecule. And even we have discussed with the different type of proteins, or you can say the terminology regarding these protein molecules. I have told you that mainly these proteins will contain the peptide bonds. So all the amino acids are jointed with each other by a peptide bond. Due to the presence of peptide bond, these proteins are said to be the polypeptides. And we have seen regarding the terminology of the proteins, I have told you minimum amount of the amino acid that has to be present for a molecule to be called as a protein is 2 amino acids. For the sugars, I have told you mainly the 3 carbon units are the minimum requirement for a molecule to be called as a sugar. So in the same way, here we have the 2 amino acids, which is a minimum requirement for a molecule to be called as a protein. If it is 2, we call it as dipeptide, 3 tripeptide, and in that way, if it is less than 10, we call it as oligopeptide, more than 10, we call it as a polypeptide. And even millions of these amino acids are linked together for the formation of the proteins. Above 100, generally it is said to be a macropeptide or macromolecule. Okay, so this is regarding the terminology of the amino acids and the proteins, and later we have discussed with the different type of bonds present in the macromolecules. Mainly in the case of 3 important macromolecules, I have discussed the bonding. One is regarding the polysaccharides. Polysaccharides will contain the glycosidic bond, and we have the protein molecules. They will be having which bond, children? It is peptide bond. And in the case of the nucleotides, if you see, the bond will be the phosphodiester bond. So all these bonds are mainly linking the monomeric units, which are the repeating units. Okay, so all are similar units which are being linked together, and hence all these 3 molecules are said to be the polymers. Even we have discussed the lipids, which are not said to be the polymers. Why? Because the repeating units are absent there. So this is regarding the last lecture. The previous video we have discussed with all these things, and later we have discussed the properties of amino acids. In properties, mainly you have to remember these 3 terms, children. What are the 3 terms? I have told you mainly this amino acid, it's an organic molecule, and organic molecule mainly containing the carbon as well as a hydrogen along with the nitrogen. So nitrogen is the most important constituent of the amino amino acid. Then what is the structure of amino acid we have discussed? The carbon, it is present, and carbon, it is mainly having the valency, it is 4. And all the 4 sides of the carbon, there are different type of chemical groups which are mainly attached to this carbon, and all these chemical groups are different from each other, and hence we call that carbon as a asymmetric carbon, and it is said to be the alpha carbon. Why? Because the remaining carbons which are attached to this carbon, it will be called as beta, gamma, based upon the number. So this is the first carbon, this is the major carbon present for all type of amino acids, and we call it as a alpha carbon. So on this alpha carbon, what are the 4 groups that are mainly present? The basic skeleton of an amino acid, for any type of amino acid, if you observe, the 3 basic groups are always similar. What are the 3 basic groups? One is the carboxyl group, which is said to be the acidic group, and we have the basic group, which is said to be the amino group, and even we have the hydrogen above, and on the left side, if you see, we have an alkyl group, that is the R group. Then how these amino acids are differentiated from each other? That is mainly based upon the alkyl group. So all the 3 groups which we have previously discussed, that is regarding the hydrogen, or you can say the carboxylic group or the amino group, all of the 3 groups are common for each and every amino acid. So while drawing the structures of the amino acids, you can make a basic skeleton of all these 3 groups, and later you will be learning with the R group, which is essentially the changing group or varying group in the case of amino acid. Okay, so the R group is mainly changing for each and every amino acid. Based upon that, the amino acids are different or unique, you can say. Okay, and later we have discussed with the properties of amino acids. There I have told you these are dipolar molecules. They are never seen uncharged. Why? Because they are acting like amphoteric molecules. They have both acidic as well as the basic properties because of the carboxylic group as well as the amino group. Carboxylic group will be providing the acidic nature to the amino acid, and the NH2 group, it is mainly providing the basic nature to the amino acid. Because the properties, because of both these properties, these molecules are said to be the amphoteric molecules. And even these molecules can show the dipolar nature, the positive and negative charge, they can be seen at a time, and such molecules are said to be the zwitterions. And later we have discussed with the properties of amino acids acting as buffering system or as buffers. So what are buffers? I have told you buffers are the organic, buffers are the chemical solutions which will resist the change in the pH. So one such important chemical molecule is the amino acid. Amino acid will be having the dipolar nature, and I have told you at the neutral pH they will be having the zwitterion component, that is plus and minus will be present together. And in the case of the acidic solution, these amino acids are acting like base, and in the basic solution they are acting like acids. So above 7, that is in the higher pH, it will be acting like a acid, and below 7, that is the pH when it is low, it is acting like, in the acidic solution, it is acting like a base. Okay, so these are the things which we have discussed in the last lecture, children, and I have discussed the importance of the buffers, and we have seen certain examples with the buffer systems, phosphate buffer and also the glycine buffers, which are mainly used in the chemical laboratories. And now today we'll be discussing with the different type of amino acids which are associated with our body functioning, that we'll be discussing today along with the classification. And in the last lecture even I have asked you to mainly study with these different type of amino acids which are present inside our body. So what are the 2 types of amino acids, the basic classification which I have given in the last lecture, children. So it is mainly regarding the essential amino acids and non-essential amino acids. Okay, so today we'll be discussing with this essential, non-essential amino acids along with the peptide bond formation. So we'll be first discussing with the peptide bond formation, later I will be discussing with the type of amino acids present in our system. So let us say this is a protein chain or a polypeptide, you can say. Here I have given the 5 amino acids which are linked together. And what are these links called as, children? These are the peptide bonds. So how these peptide bonds are formed in between the amino acid? So here in this case, all these are the peptide bonds. And how many peptide bonds we have? So same like that of the glycosidic bonds, in between 2 amino acid we will be having 1 peptide bond. So it will be easy for you for the calculation if 10 amino acids are given. obviously will be having the 5 peptide bonds. In the same way where we have discussed with the glycosidic bonds, so in the same manner we have the one peptide bond for 2 amino acids here. So in this, how this peptide bond formation it is taking place, if you observe the chemical structure or chemically how this process it is taking place, that we will see. So let us say this is one amino acid, any amino acid between 3 and 4 or 4 and 5 or 1 and 2, you will be having the same peptide bond formation. So let us say some randomly will take first amino acid and the second amino acid, they are coming together in the formation of the peptide bond. So look at the structure. So this is the amino acid 1 which is having the 4 positions of the 4 functional groups I have given here, and the same manner I have given for the second amino acid as well, that is 4 functional groups. So if you observe from in this picture here, I have given the carboxylic group, and on the other side the orientation it will be opposite, that is towards the NH2 group. So one basic and one acidic group, they will be meeting together in the formation of the peptide bond. And how this peptide bond formation or joining will be taking place here. So here there will be exclusion of the water molecule. So same like that of the glycosidic bond formation, that the dehydration reaction will be taking place. So in the same manner even we have the same reaction, that is dehydration, it will be taking place. Okay, so let us say, so in NH2 one hydrogen it will be substituted and it will be removed, and they are joined together in the formation of the water molecule. So here the water molecule it will be released, and later both these molecules they will be joined to form the peptide bond. So let us see how the peptide bond is formed here. So this is a region where the peptide bond formation has been taken place. Okay. So if you observe the structure from this diagram, the excluded molecules here are one hydrogen, it is removed from the NH2 group or the basic group of the second amino acid, and OH group will be removed from the carboxylic group of the first amino acid. So in this manner, if you observe here, there is a reaction in between the 2 amino acids where we have the formation of the peptide bond. So essentially what we are calling it as a peptide bond, that you can see here, the NH and C double bond O, all this 4 together constitute the peptide bond. So removal of one water molecule, so one for the formation of each peptide bond there will be removal of one water molecule here. Okay. So this is how the peptide bond is formed, and it is very easy for you to remember what type of chemical structure it is present in the peptide bond by remembering it as honk. Okay, so HONC I call it as a honk. So that will be easy for you to remember the peptide bond formation. Okay, so this is a kind of a code so that you easily remember the structure of the peptide bond. And now let us see how many peptide bonds are formed in this case. How many peptide bonds are seen, children? We have 5 amino acids, so we have 4 peptide bonds which are formed here. So this is how you will be calculating the peptide bond. And next to this, we'll be discussing with the type of amino acids that are present inside our body. So this is regarding the essential and non-essential amino acid list I have given here. So these are the essential amino acids and these are the non-essential. So once again, what are the essential amino acids? Amino acids which cannot be produced inside our body. They are said to be essential because they has to be inculcated in our diet. Okay, so we have to take these amino acids into our body only through the food that we eat, and hence we call it as the essential amino acids. So based upon the body requirements, this classification has been given. Then what about the non-essential amino acids? The amino acids which can be produced inside our body. The body will be having capability for the production of these amino acids, and such amino acids are said to be the non-essential amino acids. Okay. So in this we have in total of 20 standard amino acids. So the most common amino acids that are seen in any living organisms, you can say, that are mainly the 20 amino acids. Okay, so these are said to be the primary amino acids or standard amino acids, or you can also call them as the basic amino acids. Okay, so in total we have 20 standard amino acids. So what are the 20 amino acids? So these 20 amino acids, these are seen in all kind of proteins. Generally these are associated with all the protein structures. Yes, we have certain cases where some of these amino acids are absent, but still all these 20 are commonly seen in all type of the proteins. Okay, so in this 20 standard amino acids, we have divided this 20 amino acids into essential and non-essential based upon their production inside our body. So out of these 20, 10 amino acids are said to be the essential ones and 10 amino acids are said to be the non-essential category. Now let us see what are the 10 amino acids which are essential for our body, as they are not produced inside our body, they are essential. So we have the phenylalanine, valine, tryptophan, threonine, isoleucine, methionine and histidine, arginine, leucine and lysine. Okay, so for this we have a mnemonic that I have collected from the web source. Even you might have seen regarding this mnemonic. It is given as PVT Tim Hall, that is private Tim Hall. Okay, so I'm giving a mnemonic to remember. So a lot of things has to be remembered here. So mainly in the case of amino acids, we have different type of names which are new to you. So you have to remember all these names with the help of some codes. So I have designed some codes regarding the polar and non-polar amino acids. So that will be discussed later. So now we'll be writing some codes for these amino acids under essential and non-essential category. Okay, so we have a code for this, that is private Tim Hall. Okay, so here the PVT stands for phenylalanine, valine and tryptophan. Okay, so these three, it will be coming under the phenylalanine, valine and tryptophan. And next we have Tim, that is T-threonine, isoleucine and methionine. Okay, so these three will come under the Tim. And HALL Hall, it will be coming along with the histidine, arginine, leucine and lysine. Okay, so these are the set of amino acids, and you have to remember this, all these 10 amino acids will be coming under the first category. And coming to the non-essential amino acids, we have other 10 amino acids which are listed in the non-essential category. And in this you have the similar amino acids with the letters which are starting with the similar letters will be present for all the amino acids. More common, A and G will be present. So what I have done, so I have written all the amino acids starting with A in the first order and with G in the second order, and the last one we have some variable letters. So what we will do, we will write a code here. So A cube, G cube, and here it is CST P. Okay, so I will tell you what is this code here. So all of you have to remember this. So A3 means alanine, asparagine, aspartic acid. So all the first three amino acids under non-essential category, these are produced inside our body. So we have the ability to produce these amino acids. So alanine, asparagine and aspartic acid, that will come under A cube. And we have glycine, glutamine and glutamic acid. So glycine, glutamine and glutamic acids, all these three will come under G cube. Okay, and lastly we have the CSTP. So for this I have made a code here. So CST, we all know the Central Standard Time. So here you have to remember the Central Standard Time for Peru. Okay, so for Peru I have given the Central Standard Time for Peru. So there you have the C is for cysteine, S is for serine, T is for tyrosine and P, lastly the P is for the proline. So let us see here. So we have this code A cube, G cube and CSTP. A cube again, once again if you see all the three amino acids are in A. Then the problem is that there are some amino acids which have the common letters in the essential amino acid category. Then how you will differentiate? Let us see. So what are the letters that are matching with the essential category here? So with A, we have three amino acids in the non-essential category. So in this essential category, if you see, we have only one amino acid. Okay, so this is one amino acid which is starting with A. Then how you will differentiate? So for this again, we have a small technique here. Out of these 10 amino acids, the histidine and arginine, they are said to be the semi-essential amino acid. So these two are said to be the semi-essential amino acids. Okay, so I'm making a star mark over here. Histidine and arginine are said to be the semi-essential amino acids. Why they are said to be the semi-essential ones? They can be produced inside our body, but it is very slow process, or you can say rarely you can see the formation of these amino acids, and hence we have to take them even in our diet. Okay, so we have to include the proteins which are having these amino acids in our diet. Not only that, during certain times, like you can see the conditions like trauma or the adolescence or during the pregnancy, these two amino acids, they can't be produced inside our body. That will make it important to categorize under the essential amino acids. So in this way, if you remember that arginine is an semi-essential amino acid, so only two we have in the special category, that is histidine and arginine. So you have to by heart these things. Histidine and arginine, they will come under the semi-essential category, though they are included under essential. You have to remember they are also a part of special category. During certain conditions, these amino acids, they cannot be completely produced inside our body. Okay, so these two if you exclude, in that way if you see, other three amino acids starting with A will fall under the non-essential category. Okay, so the first important thing here, the first challenge it is solved here. Then what about the other amino acids which are having the matching names here? Let us see, matching letters here. So we have G here and we don't have any G here, so you can simply remember the G cube means the amino acids which are starting with the G, they will be completely present in the non-essential category. And what about the other four we have? We have cysteine, we don't have any amino acid starting with the C here, there's no problem. And we have serine here, again we don't have any S starting with S. And next is about the proline, if you see P here, we have P here, so phenylalanine. And here we have proline, so both are having the same name here. And also the tyrosine, it is starting with T here, and even we have the two other amino acids starting with T here. So the problem lies in these two amino acids to remember. So there it is tyrosine and proline, and here it is phenylalanine, tryptophan and threonine. So how you will remember these amino acids again? So in this category, if you see the last order which I have given, the CSTP, this is for all these four amino acids. In this you have to make a notice that the first two letters of the cysteine, it will start with CY. And hence you have to remember that the letter starting with the amino acid starting with the T even will have Y here. So what it is, if it is having TY, it will be the tyrosine. So in that way you can solve this problem. Okay, so CY along with TY it is present. Then what about the proline? So what we will do with the proline, let us see. So for proline, what you will do, children, again remember the same thing. So what I have coupled two amino acids of these four. So I have coupled the cysteine with tyrosine, so you can easily remember that CY will be for cysteine and TY is for tyrosine. And the same thing you will be, if you can see the similarity between the serine and the proline. Even in the serine we have SR, the third letter SR. In this case again we have PR. So in this way you can easily remember this. Okay, so cysteine and tyrosine, they will have a similar match, and serine and proline, they will be having a similar match. So in this way you can make an easy code out of this. Okay, we have the PVT, Tim Hall. So PVT stands for phenylalanine, valine, tryptophan, Tim for threonine, isoleucine and methionine, and Hall is for histidine, arginine, leucine and lysine. So this you have to remember in this way. And in this, essentially, you have to remember the histidine and arginine are said to be the semi-essential amino acids. Okay, So this is regarding the 2 amino acids. And coming to the non-essential category, we have A cube, G cube and CSTP, Central Standard Time for Peru. Okay, and in this A cube I have given alanine, asparagine and aspartic acid. Even we have similarity here, alanine, glycine, asparagine, glutamine, aspartic acid, glutamic acid. Even these 2 categories are very similar, so it is very easy for you to remember. And last one, we have the CSTP, where we have faced a problem that is regarding the similarity issue of these amino acids with the essential amino acids. So that I have solved for you. Cysteine, it is similar to thyroxine, TY, CY, and serine with the proline, SR, and we have PR here. Okay, so this is how you will remember the essential and non-essential amino acid category. Next, coming to the, so we have learned that the 20 standard or primary amino acids are seen in any type of living organism. Then what about the other amino acids? So other than this, we have different categories of amino acids that I will be giving certain examples here. So look at the other amino acids. We have more than 300 amino acids which are present inside the living organisms. So other than this 20, these 20 are the primary amino acids which are making all the proteins. So all the proteins will be having in common all these primary amino acids, and we'll be learning in +1 and +2 regarding these 20 standard amino acids. It is not required to learn all the non-standard amino acid, but still you should have an idea regarding this non-standard amino acid. Why? Because in certain cases you will be learning with the certain terms, so that it will be easy for you to learn or easy for you to be aware that those substances are nothing but the amino acids. Okay, so we have certain amino acids, non-standard amino acids, which will be discussed in bacteria, even in the blood clotting factors. They are mainly associated with the non-standard amino acids. So let us see the familiar examples which are mainly related to your study that I will be discussing here. So non-standard amino acids are rarely seen or associated with only certain kind of proteins. Not only that, sometimes these non-standard amino acids, they are freely present in the cytoplasm. So based upon that, we have 2 categories here. Non-standard protein amino acids which are associated with the protein. So along with the proteins, along with the 20 standard amino acids, they will be present as a combination inside the protein, and such amino acids are said to be the protein amino acids. And some amino acids, they are not a part of protein, but they are present freely in the cytoplasm. They are not jointed to the protein molecule, but still they are amino acids. They are said to be the non-protein amino acids. Now let us see. So what are the important examples that you have to study here? So in this category, firstly we'll be discussing with the hydroxy lysine and hydroxy proline. So lysine and proline, both are normal amino acids, the primary amino acids, and these are modified with the extra hydroxyl group, and they are said to be the hydroxy lysine and hydroxy proline. If you see these 2 amino acids, they are associated with a protein called collagen. So mainly, in the universe, the most abundant protein for animal kingdom is the collagen, and the most abundant protein for the plant kingdom, it is the rubisco enzyme. We all know it is mainly seen in the case of the photosynthesis. The main enzyme in photosynthesis is the rubisco. So that rubisco, it is mainly seen in the plant kingdom, but for animal kingdom we have the collagen. So this collagen protein, along with the other amino acids, it is associated with the other non-standard amino acids which are modified amino acids, and those are hydroxy lysine and hydroxy proline. Mainly this hydroxy proline, it is more compatible with the lysine. And another example, we have the carboxy glutamate. It is a part of the blood clotting factor, prothrombin. We all know prothrombin is a blood clotting factor, and that is mainly made up of a modified glutamate. It is mainly, we all know the glutamate is the glutamic acid, and this is modified along with the carboxy group, and it is said to be the carboxy glutamate. It is included in the blood clotting factor, the thrombin. And next we have other important thing, that is alpha epsilon diaminopimylate. So this we will be discussing in the bacteria section, children. So in bacteria we have some endospores, and endospores, they will be having certain substances which will help in giving resistance to the bacteria against the stress, and such substances are mainly the diaminopimylates, mainly seen in the case of endospores, that we will see in the bacteria we will be discussing. And this diaminopimylate, it is mainly seen in the bacteria. Okay, so these are the most important examples that you have to remember. These are amino acid, but still they are said to be the non-standard. Why? Because they are rarely seen in the case of the living organisms, and they are associated only with a specific type of proteins. And coming to the non-protein amino acids, so these are completely free in nature. So free means they are present freely in the cytoplasm, and they are never associated with the protein molecules. Such amino acids are said to be the non-protein amino acids. So in this we have certain examples, children. So citrulline, ornithine, all these are the non-protein amino acids. They are never associated with any kind of the protein. They are freely present in the cytoplasm. If you see these examples, they are mainly associated with the urea cycle of animals. Okay, so that will be discussing later. So these are certain examples which are mainly related to your study, and hence I have given this. So these are non-standard amino acids. More than 300 amino acids are coming under this category, non-standard amino acids. So where again, based upon their association with the protein, they are said to be the protein amino acids. If they are not associated, if they are freely present, we call it as the non-protein amino acids. Okay, so these are the list of examples which I have given. And next, let us move on to the structure of the 20 standard amino acids. So we are not going to discuss with the structures of the non-standard, it is not required. We will be mainly concentrating on the primary amino acids and their structures. Along with that, you have to know the chemical nature of the amino acids. So what type of amino acids are present inside our body based upon the alkyl group. I have told you in common the amino acids will be having all the three functional groups which are similar. So that will make the basic skeleton of the amino acids. Then what is making them different or unique? That is mainly dependent upon the R group, that is the alkyl group. So for each amino acid will make a basic skeleton. Even if you are preparing a notes, what you will do, you will make a basic skeleton for all the amino acids. 20 basic skeletal structures you have to make, that is the alpha carbon in the center, and on all three sides we have the H on one region, and other you have the carboxyl group, and on the other side you will be having the amino group. So all these three will make the basic structure for the amino acid, and later we have one extra group, that is the alkyl group, that you will leave a gap. So what will happen? So it is very easy for you to remember. So all the R groups will be written on one side. So that will be, in this way you can learn the structures of the amino acids. Okay. So while discussing with any type of structure, where mainly we are calling them as polymers, that you have to remember regarding their directionality. So in what direction? If there are a lot of amino acids, so when you are studying certain amino acids, then how you will be naming them? In what direction you will name them? That is very important, which is said to be the directionality. So in the case of even the nucleotides or the carbohydrate, the sugar units, you will be following certain rule in studying, or you can say in writing the structures of these molecules. In the same way, if you see the directionality of the protein or amino acids, the directionality means we will label each end of these protein molecules. The proteins are made up of numerous amino acids. If you see, this is like a chain, and never a protein can exist as a straight chain, children. Simply for our understanding, we are simply writing that we are representing them in the form of a straight chain compound. And here all the amino acids are linked with the peptide bonds, and all the amino acids will be having two major ends which are free. So once the amino acids are joined, forming a protein molecule, the two sides will be free. What are the two sides, children? One, we will be having the left region, and other we will be having the right region. And the left region, where mainly you can see the free NH2 group or amino group, it is said to be the N-terminus or N-terminal, and the other group which is present towards the right side, it is said to be the C-terminus or C-terminal because of the presence of acidic group, which is a COOH or carboxylic group. So always you have to remember, you have to follow certain rules in studying and representing these molecules. Why? Because numerous type of linking units or repeating units are present here. So always while writing the amino acids, you have to follow this standard rule. You have to write the amino acids starting from N region to C region. So let us say we have amino acid threonine in the N region. So the first amino acid, it will be definitely the threonine. So while writing, you have to start reading the amino acids from this region to the C-terminus region. It is always, the directionality is important here in studying as well as in the representation. It is always from N to C. Okay, so this you have to remember. All these amino acids which are starting, even in your exams, you will be getting some structures which are having the N-terminus and C-terminus. Then you have to remember that even it is given in either right or left, you have to follow N to C rule. Always read the amino acids or write the amino acids from N-terminus to the C-terminus. Okay, so the amino acid which is present towards the N-terminus will be the first amino acid, and towards the C-terminus will be the last amino acid. Okay, so this is one important thing you have to remember. And coming to the structures of the 20 standard or general amino acids. So these are the 20 amino acids or general, you can say, you can call them as a primary or the 20 standard amino acids. So all these 20 standard amino acids, they will be having different properties. But what I have told you, I have told you that it is having some carbon skeleton which is common to each and every amino acid. The functional groups, there are certain functional groups which are common to all the amino acids. So in this again, in all these chemical structures, if you observe, there are certain square boxes which I have made, which means the amino acids which are having the common skeleton. So all these functional groups are common to all the amino acids without missing any one of the amino acids. If you see from starting from one to the last one, so all these amino acids will have something in common that we will see. And what is changing in them? So whatever groups that I have written other than or behind the box, it will be mainly the R group or the alkyl group. So that alkyl group will make the difference here. Okay, so it is not important for you to learn all these structures, children. It is not important here, mainly even for your NEET or other exams. It is not essential to learn the structures, but you have to know what type of amino acid will be present in which type of category. So the amino acids are mainly modified with different type of compounds. So all these addition compounds that you have to remember. It is very, very important even for your NEET or other exams. But remember, what you have to learn is the categorization. So we have different categories of amino acids based upon their chemical structure. So additional chemical groups, based upon that different categories were given here. All these categories that you have to that you have to study mainly. Okay, so here what are the different type of categories we have, that first I will be explaining here. So we have 8 categories of amino acids. So what are the 8 categories we have? So the first being the acidic amino acids. Amino acids are having the carboxylic group and hence they are said to be acidic. And other than this additional group, so other than this carboxylic group, we have another additional carboxylic group which is making the amino acid more acidic, and such amino acids are said to be the acidic amino acids. In this category, we have 2 amino acids. What are the 2 amino acids, children? Aspartic acid, the name itself indicates it is an acidic amino acid, and glutamic acid. So look at the structure here. These are the common functional groups present for these acidic aspartic acid and glutamic acid, which I have represented in the red. So I am not discussing with all the common groups every time. So you have to remember, if this red zone is given for the amino acid, which means it is common to all the amino acids. Okay. So apart from this, we'll be discussing. So what additional group it is present? We have in general the CH, COOH and NH2. Okay, so here the three functional groups are hydrogen on one side, carboxylic group on one side and amino group. Other than this, we have in acidic amino acids, we have the carboxylic group. One extra carboxylic group will be present which make them more acidic, and hence they are said to be the acidic amino acids. Under this category we have the aspartic acid and glutamic acid. These are the two acidic amino acids. And next, coming to the basic amino acid. So the name itself indicate it will be having an additional basic group. What is a basic group generally present for amino acid? We have NH2 group. Amino group will be present. So this is the common group. Amino and carboxylic, it will be common. And other than this NH2 group, we have additional NH2 group which make these amino acid more basic. And if you look at the structure where you have the NH2 group here, so we have in the last zone. Okay, so this is the NH2 group. So because of the presence of additional NH2 group, these are mainly said to be the basic amino acid. So here I have not mentioned, children. So the first amino acid, which is the basic amino acid, is the lysine, and the second amino acid, it is the arginine. Okay, so these are the two basic amino acids having one additional amino group. So looking at the structure, it will be easy for you to understand, and hence I have given a complete structure of these amino acids. But remember, it is not required for you to learn all these structures. Simply you have to know the categories, the amino acids which will fall under these categories. So all these categories you have to learn and the names which are present inside it. Okay, and next, coming to the arginine, here even we have the additional amino groups are present. NH and NH will be present, which will make it more basic. So that is arginine. And next, coming to the sulfur containing amino acids. So under this category, the amino acids which have the sulfur as additional group, we call them as sulfur containing amino acids. So under this we have cysteine and methionine. Okay, cysteine and methionine will be coming under the sulfur containing amino acids. These amino acids contain the sulfur. SH here and here S it is present. Okay, sulfide hydride is present here. And next, coming to the alcoholic amino acid, the name itself indicate they will be having the OH group present in them. So here the two amino acids which will have the alcoholic groups, which are said to be the alcoholic amino acids, are one the serine and two is the threonine. Serine and threonine both are said to be the alcoholic amino acids. Why? Because they have the OH groups present in them. So okay, so these are the two OH groups present in them and they are said to be simply the alcoholic amino acids. And next, coming to the simple amino acids or neutral amino acids. Simple or neutral means mainly they will be having, why they are said to be neutral? Why? Because they have one amino group and one carboxylic group, which means one acidic group and one basic group. So both will balance and it will give a net, the charge will be zero. That is the neutral amino acid category. So mainly these 5 amino acids will come under the simplest amino acids, or simply these are the simple amino acids, or they are said to be the neutral amino acids. So in this again, the glycine is the most simplest form of amino acid. So I have told you glycine is mainly used as buffering system or buffers. So if you look at the structure, other than the common structure, we have one additional hydrogen. So this is the simplest structure that even you can learn this. Okay, so this is the structure of the glycine. Other than glycine, we have leucine, isoleucine, alanine and valine. All these 5, glycine, leucine, isoleucine, alanine and valine, all these 5 amino acids will be coming under which category, children? Simple or neutral amino acids. For each and every amino acid, if you observe, there will be one acidic group and one basic group, COOH, NH2, COOH, NH2. Other than that, we have only methyl or methylene groups will be present, but we don't have any additional groups or additional compounds which are attached to them, and hence they are categorized under the simple or neutral amino acids. Okay, so this is one type of category we have. And coming to the next category, that is aromatic amino acids. The amino acids which will release aroma, or you can say the odor, we call them as the aromatic amino acids. Even when you're experimenting with these amino acids, you can feel the fruity smell or fruit smell in the laboratories, and these are mainly the phenylalanine and tyrosine. Okay, so phenylalanine here, if you observe, as they are releasing some aroma, they are included under the aromatic amino acid, but the structure it is slightly different. If you observe, one, the tyrosine, it will be having the hydroxyl group, and even the ring structure will be present, but due to the presence of, we have another group of amino acid where the rings are seen, and those are included under heterocyclic amino acid, where the different ring structures or rings are present, but these are not included in them because of their specific nature of releasing aroma. They are again differentiated and they are put into the category of the aromatic amino acid. Okay, so phenylalanine and tyrosine both are coming under which category, children? Aromatic amino acids. And lastly, we have the two amino acids here, groups here. One is heterocyclic amino acids. So different ring structures are formed here, and hence they are said to be the heterocyclic amino acids. So let us see what type of cyclic structures are seen in these amino acids. The first one is proline. So proline, histidine, tryptophan. Once again, proline, histidine and tryptophan, all these three are coming under the heterocyclic amino acids. And in this, again, the tryptophan is also the aromatic amino acid. So how many amino acids which are aromatic here? We have phenylalanine and tyrosine, but along with them we have the tryptophan. But tryptophan, it is not included under the aromatic category. Why? Because they mainly have the heterocyclic nature. Heterocyclic means they have different rings here. One, you can see the 6-membered ring, and other they have the 5-membered ring. So this makes the difference mainly because of their heterocyclic nature. The tryptophan, being aromatic, is present in the heterocyclic group. Okay, so but you have to remember that there are 3 major amino acids which are mainly the aromatic amino acids: phenylalanine, tyrosine and tryptophan. Okay, so these 3 are the aromatic amino acid. Which amino acid is said to be aromatic and heterocyclic? Then you have to mark it as the tryptophan. Okay, the questions will be asked in this manner, children. If they are asking for aromatic amino acid, all these 3 will be coming under the aromatic category. If they are mainly asking for aromatic and heterocyclic, then it will be the tryptophan is your answer. Okay, so this is regarding tryptophan. And other than that, we have histidine and even the proline. So here even you can see the ring structure, that is 5-membered ring, it is mainly seen, and these 2 rings are present in common for both proline as well as histidine. So all these are the ring structures, and mainly the tryptophan, it is having both the heterocyclic nature and also the aromatic nature, and hence they can be categorized under both categories. Even you can keep the tryptophan under aromatic category because of its aromatic nature, or it can be placed under the heterocyclic category because of the presence of ring structure. Okay, so essentially mainly it is having the cyclic rings, or you can say they are forming the ring structure. The straight chain conformation, it is again having the ring form, and hence it is said to be the heterocyclic. In some we have 2 different type of rings, and in certain amino acids only one ring can be seen here, the proline and the histidine. Okay, so this is regarding the heterocyclic amino acids. And lastly, we have the 2 more amino acids which are carboxy amino acids. Carboxy means we have the CO group, which is additional group present in these amino acids, and under this category we have 2 amino acids, children. One I have not given the name, asparagine and glutamine. Okay, asparagine and glutamine, they are said to be the carboxy amino acids. Okay, let us write it. Okay, so this is the last category of amino acids. We have carboxy amino acids, where you can see the C double bond O, which is mainly present in these amino acids. Okay, due to the presence of this carboxy group, these amino acids are said to be the carboxy amino acids. Under that we have asparagine and glutamine. Okay, So this you have to remember each and every category and the amino acids which are falling under this category, that has to be memorized. Okay, here we don't have any tricks here. Simply by looking at the structures, if you make a notes with these structures, it will be very easy for you to remember. By looking at the pictures, it is very easy for you to remember the structure and their nature, which type of chemical compounds are mainly being added, that can be compared and can be studied easily. Okay. So this is regarding the 8 groups of amino acids. So all the 20 amino acids we have here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, so all the 20 amino acids are grouped under this 8 categories. Okay, and other than this, we have another classification that is the most important for you regarding your NEET. Some amino acids are said to be the polar amino acids, and some amino acids are said to be the non-polar amino acids. So that we will be discussing in the next lecture. So today we will be mainly discussing with the type of amino acids. I will be giving a quick review of all these amino acids here. So what are the groups we have? The first group which I have discussed, it is regarding the acidic amino acid. Acidic amino acids, that examples are, it is very easy because the name itself having this acid in their name. Okay, aspartic acid and glutamic acid, they will come under the acidic amino acids. And next we have discussed with the basic amino acids. Basic amino acids, they are a part of histone proteins. We have discussed in the DNA. The lysine and arginine, both are the basic amino acids because they have one additional basic group, that is amino group. And next we have discussed with the sulfur containing amino acids. Sulfur containing amino acids are cysteine and methionine. Both these amino acids, they will be having the sulfur as an additional group. They are included under sulfur containing amino acid. And next we have the alcoholic amino acids. Alcoholic amino acids, they will be having additional hydroxyl group. And what are the amino acids we have under this category? The serine and threonine. Serine and threonine will come under the alcoholic amino acids. And next we have the aromatic amino acids. In this category I have discussed with the 2 amino acids, but mainly you can even include the tryptophan under this. So what are the 2 amino acids which we have discussed, children? We have the aromatic amino acids which are phenylalanine, and we have tyrosine which we have discussed, even we have the tryptophan. Okay, so phenylalanine, tyrosine and tryptophan, all these 3. Yes, you can learn this tryptophan under aromatic, or you can even learn them under the heterocyclic. That is of your wish, but you have to remember that it is common for both. They have the both the properties are present for the tryptophan. And next we have discussed with the simple amino acids or neutral amino acids. They will be having no additional groups here. One acidic and one basic group. It will give the net 0. That is, the charge will be balanced here, and hence they are said to be the neutral amino acids or simple amino acids. In the simple amino acid, the simplest amino acid is the glycine, and the first discovered amino acid is the leucine. Okay, so what are the 5 amino acids that are coming under this category? We have the valine, the leucine, isoleucine, glycine, and also we have the last one, alanine. So all these 5 amino acids will be coming under the simple amino acids or neutral amino acids. And lastly, we have discussed with the carboxy amino acid and heterocyclic amino acid. The carboxy amino acids will be having the carboxy group, and under this category we have discussed with 2 amino acids, asparagine and glutamine. And lastly, we have discussed with the heterocyclic amino acid. Don't get confused with this name, children. Simply remember cyclic amino acids. Cyclic amino acids means they will be having the cyclic structures. Other than the straight chain, they will be having the ring structures in them. And what type of amino acids are coming under this category? We have proline, we have histidine, and we have tryptophan. Tryptophan, it is mainly having the 2 rings, that is the heterocyclic rings are mainly seen in tryptophan along with the aromatic property. And for proline and histidine, the single ring will be present. So this is regarding the 20 standard amino acids which are mainly included as a part of the proteins. So proteins are different type of proteins are present, but mainly remember all the 20 amino acids are generally present in the proteins, but there are some proteins which lack some of the essential amino acids which are required for our body. So in the next lecture I'll be discussing with the type of proteins which are having the amino acids and also regarding the non-polar and the polar amino acids. So while coming to the next lecture, it will be very easy for you if you learn all these different classifications I have given in this lecture. So one, we have learned with the essential and non-essential amino acid. What are the amino acids that are coming under the essential category? So first I have told you the phenylalanine, the private TIM hall that you have to remember, the code which I have given, the PVT TIM hall. P is for phenylalanine, valine, and we have tryptophan. And next in that, the second category, the TIM, it will include the threonine, and isoleucine we have methionine, and hall it will include the histidine. A is for arginine, and L for leucine and glycine. And in this I have told you the histidine as well as the arginine, they are said to be the semi-essential based upon their production or the nature of their production inside the human body. And next we have discussed with the essential amino acids. Essential amino acids are mainly having a code which I have given for you, that is the A cube, G cube, and also CSTP. CSTP means Central Standard Time for Peru, that I have told you. So A means alanine, asparagine, and also we have discussed with aspartic acid. G means glutamine, glutamic acid, and also glycine. And lastly we have CSTP, that is mainly for cysteine, tyrosine, which are similar, and serine and proline, which are similar again. So that is what we have discussed in essential, non-essential, and we also discussed with the non-standard amino acids, which are not important for you, but it is still important to remember these names. Why? Because while discussing with something which is related to you, then it will be very easy for you to understand that these are also coming under the amino acid category. Okay, so this is what we have discussed today, and while coming to the next lecture, I expect you to learn all these terms, mainly the 20 amino acids you have to by heart and come, so that it will be very easy for you to learn about the protein structures and also regarding the different type of amino acids, the non-polar and polar amino acids. Thanks for watching. Please subscribe my channel. Thank you.